Cells can signal to each other and interpret the signals they receive from the other cells and the environment. So in here, there is one cell which gives the signal, which sends the signal, and there's another cell which takes the signal or which receives the signal. Okay, so these are first two elements for signaling. So cell signaling has four elements. The first one is the signaling cell, the signal producing cell. The second one is the target cell, signal receiving cell, the cell receiving the signal. Because from the four elements, there are the first one, signal emitting cell. Especially I'm using different terms, which means the same thing. Signal producing cell or signal emitting cell. These are both the same. Or signal sending cell or signaling cell. These are all mean the same thing. So one cell produces the signal. The other cell is the target cell or signal receiving cell. The cells can receive the signals also from the environment, from their environment. We will see examples in a few minutes. Signals are most often chemicals. The same small set of cell signaling mechanisms shows up in diverse species and processes. So, similar or same signaling mechanisms done by similar molecules or the same molecules are seen in different species. So, for example, in this picture, you can see two different animals. So, one of them is trying to hunt the other one. Actually, at the moment, when this picture taken, at that moment, the picture is taken, both of the animals are using the same signaling mechanism, same cellular signaling. Now you will say, okay, these are both very large animals, but you are talking about the cell signaling. That's right. Both of these animals move, or the thing make these both animals move or show these behaviors are or is the cell signaling, the cellular signaling. How does it work? Because both of them at the moment use the epinephrine mechanism. Both of the animals here at that moment have the same hormone, same endocrine, epinephrine or adrenaline. Maybe you are more familiar with the name adrenaline. Its other name is epinephrine. It's a hormone. And this is the molecular structure. This is the space-filling model of epinephrine. Okay, how does it work? External signals are converted to responses within the cell. Communication among microorganisms provides some insight into how cells send, receive, and respond to the signals. Now, here, response, cevap demek. Aklınızda bulunsun. Response, cevap. Send, göndermek biliyorsunuz. Receive, almak, algılamak. Evolution of the cell signaling. So let's look at the evolution of cell signaling. The cell signaling is seen even in microorganisms, even in bacteria. Even in bacterial cells have cell signaling mechanisms. The example here is the yeast, Saccharomyces cerevisiae. So the yeast have two types. Mating types, we call them mating types, A and alpha. So these are single cell organisms. Some of them, even though they are the same species, so from this species, some of them are A type, some of them are alpha type, just like female and male. Cells of different mating types locate each other via secreted factors specific to each type. Secret, salgılamak demek. Locate, yerini belirlemek. Evet, burada secreted, to secret, salgılamak. The binding of a mating factor. So there are two types. There are two mating types of yeast. Some of them are A, some of them are alpha, and they produce different kinds of signal molecules. They produce different kinds of signal molecules. So the A type produces A signaling molecule. The alpha type cells produce alpha signaling molecules. And A cells can receive or sense the presence of alpha type molecules, and alpha type cells can sense or receive and respond to the signals produced by A-type cells. The binding of mating factor at the cell surface initiate a series of steps called signal transduction pathway. So in here, our word, a very important word, signal transduction pathway. Signal transduction and pathway. We will use those two words a lot. Molecular details of signal transduction in yeasts and mammals are very similar, even though we are at the two different ends of eukaryotic evolution. Mammalians and yeast use very similar mechanisms, but the important difference is in mammalians there are many different signaling mechanisms, many signaling pathways, but in the yeast there are only a few signaling pathways. Now, these are our yeast cells. So this is one cell, this is another cell, this is one yeast cell. This is remember again, to remind you, yeasts are the single cell organisms. So this is the whole organism. This is a yeast cell in A type. This is a yeast cell in alpha type. A cell produces A factor, alpha produces alpha factor, and alpha factor binds to A cell, A factor binds to alpha cell. What happens? So after that, the alpha cell understands that there is an A-type cell nearby, and it moves to that, and they mate. And a new cell is an A alpha cell. But this is seen only in single cell organisms. Don't get confused. So this is one of the simplest ways of signal transduction. There is another mechanism of signal transduction which is seen in bacteria. Even though they are prokaryotes, they are even not eukaryotes. Even in the bacteria, there are signal transduction mechanisms, which is called quorum sensing, or one of the example is quorum sensing. This is an ancestral signaling system. Ancestral signaling molecules likely evolved in prokaryotes and single cell eukaryotes and were adapted for use in their multicellular descendants. The cell signaling is critical among prokaryotes. So in prokaryotes, we can see cell signaling mechanisms as well. A concentration of signaling molecules allows bacteria to sense local population density in a process called quorum sensing. An example of quorum sensing is formation of biofilm. So biofilm is an activity of bacterial cells. Biofilm is an aggregate of bacterial cells adhered to a surface. Another example is medically important: secretion of toxins by infectious bacteria. Interfering with signaling pathways used in quorum sensing may be promising approach as an alternative antibiotic treatment. So quorum sensing is the signaling mechanism seen in the bacterial cells. Now, we can go ahead, local signaling and long distance signaling. Local signaling is the communication between the cells which are close to each other. Cells in a multicellular organism communicate via signaling molecules. We know this. The local signaling in the local signaling animal cells may communicate by direct contact. In this way, the cells can directly be in touch with each other, and with this direct contact, they can affect each other's behaviors, and they can send the signals. Animal and plant cells have cell junctions that directly connect the cytoplasm and the cytoplasms of adjacent cells. So now we are talking about the local signaling and direct contact. So these cells are directly in touch with each other because they are living next to each other. They share their membranes touch each other, and they have cell junctions. You should remember from last week. And through these cell junctions, they can send directly the molecules to each other. Signaling substances in the cytosol can pass freely between adjacent cells. But these are adjacent cells, komşu hücreler. Adjacent, komşu demek, birbiriyle yan yana bulunan birbirine yapışık hücreler. All right, this is the first type of local signaling, direct contact. For example, gap junctions. I guess you can remember gap junction from last week or week before. These gap junctions are channels between adjacent cells, and through these channels, the molecules, signaling molecules, can pass from one cell to another cell. And in the plant cells, there are some structures called plasmodesmata, and the signaling molecules can pass between adjacent cells. But there is another mechanism of local signaling. That is cell-cell recognition. So these are two direct contact mechanisms. Okay? So the local signaling have different mechanisms. First group of mechanisms are direct contact. So direct contact can be with junctions, cell junctions between adjacent cells, or cell-cell recognition. The cells can bind to each other with the membrane proteins. So one cell has the first kind of membrane protein, the second cell has the second kind of membrane protein, and these two membrane proteins recognize each other and they bind to each other. This makes two cells to bind to each other. There's a question, Talip Yıldız. Hocam burada vakoller yardımcı oluyor değil mi? Evet Talip Yıldız, gap junction'lar aracılığıyla vakollerin geçişi olabilir. Veya doğrudan doğruya vakol şeklinde olmasa bile moleküller direkte geçebilir. Alright. So these are two types of local, or these are two types of direct contact between cells, between two cells. The second local signaling mechanism is paracrine signaling. In the other cases, animal cells communicate using secreted messenger molecules. So the message producing cell secretes as a signal molecule. So it releases the signaling molecule, and this molecule travels only short distances. So like in the same tissue. Growth factors which stimulate nearby target cells to grow and divide are one class of such local regulators in animals. This type of local signaling in animals is called paracrine signaling. So, for local signaling, we can say there are two mechanisms, two main mechanisms. The first one is this one, direct contact between cells. And the second one is paracrine signaling. The third one is synaptic signaling. Now it's three. So for local signaling, we can say direct contact between cells, paracrine signaling, and synaptic signaling. So did you understand? Synaptic signaling is seen in animal nervous system when a neurotransmitter is released in response to an electric signal. So in here, the synaptic signaling is seen in nervous cells, neuron cells, and in neuron cells, there are special signal molecules which are called neurotransmitters, and they are released in response to an electrical signal. And these neurotransmitters carry the signals between neural cells, nervous cells. And keep in mind, neuron cells are seen only in animals. No other kingdoms carrying neural system. Local signaling in plants is not well understood beyond communication between plasmodesmata.